Chapter 23 starts a new section of biochemistry, where we look back at the three major biomolecules, carbohydrates, lipids, and proteins, and try to understand how they're used for energy and synthesized in our bodies. Because the end parts of metabolism, the citric acid cycle and the electron transport chain, are generally shared when breaking down all three biomolecule types, we'll talk about these final and central parts of metabolism that begin with a molecule acetyl-CoA before we talk about how we get to acetyl-CoA from each type of biomolecule. In this chapter, we introduce the concept of metabolism, really just any of the reactions that our bodies do. Sometimes the reactions will be part of processes that take energy to make larger molecules that store energy. Can you think of an example of a large carbohydrate or lipid molecule that we might use to store energy? We call these processes that make larger molecules anabolic pathways. When you break down a large molecule to use the energy, this is called catabolism, and the sets of reactions that accomplish them, catabolic pathways. A pathway sometimes is linear, where we completely convert the starting material to a product, or sometimes they're cyclic, where the set of reactions give us back the original starting material, but convert a few atoms into energy or waste. Examples include the citric acid cycle and the urea cycle, or in plants, the Calvin cycle. Metabolism happens in several areas of the cells, and sometimes this leads to important features of metabolism. Most DNA metabolism occurs in the nucleus, as we saw in the last chapter. Most protein anabolism, remember this is building up a protein from the amino acids, occurs in the ribosomes, which are scattered throughout the cytoplasm of a cell. We also saw this in chapter 22. When we talked about proteolytic enzymes in chapter 21, we talked about how most protein degradation, which is catabolism, that occurs within a cell occurs in the lysozyme. Most of our central metabolism occurs in a specific organelle within the cell called the mitochondria. Mitochondria have a structure that is an outer membrane and a separate inner membrane. The inner membrane holds most of the enzymes responsible for the electron transport that will harvest energy to be used to make ATP, including ATP synthase. Between these membranes, there's an inner membrane space. The inner membrane has all sorts of folds called cristae. Why might the cristae be important to mitochondrial efficiency? The cristae increase the surface area of the inner membrane and allow for more enzymes in the same space. There are several molecules that we'll see turn up over and over and over again through the next 4 chapters. These molecules are some of the most important ways that energy is developed and used within a cell. Whenever we see these molecules, we want to pay particular attention because they help us to understand when energy is being used and when it's being produced, and how the pathways are connected. We'll talk about each group of molecules separately, then return to this summary slide. The first set of molecules are the ribonucleotide phosphates. Notice that these are simply ribonucleotides with different amounts of phosphate. The ribonucleotide portion gives an easily recognizable structure that an enzyme can bind onto. The phosphates are the major way that the energy is stored in our cells. Any ribonucleotide can store energy, but adenosine is by far the most common. Adenosine triphosphate, or ATP, is the major energy currency of cells. We're going to think about all of the energy that we talk about in ATP equivalents. Whenever ATP energy is spent, it loses a phosphate. It is most common to lose a single phosphate, which would give us adenosine diphosphate, or ADP, which we think of as the equivalent of spending 1 ATP. If we lose 2 phosphates, which is less common, we get adenosine monophosphate, AMP. We consider this as equivalent to spending 2 ATP. The next set of molecules are the flavins, flavin adenine dinucleotide, or FAD, and dihydroflavin adenine dinucleotide, FADH2. The general form only differs at the flavin, which you can see shown at the bottom, and the rest of the molecule is just to give an enzyme something to recognize. These molecules are related by redox. FAD is the oxidized form, and FADH2 is the reduced form, which means that these tend to be partners to other redox reactions on molecules. You can see the 2 forms of flavin again at the bottom. Because flavins are able to exchange 2 hydrogens at once, they are most likely cofactors for all reactions converting alkanes to alkenes, where FAD is used and FADH2 is produced, or alkenes to alkanes, in which case FADH2 is used to reduce the alkene to an alkane and FAD is produced. The nicotinamides are also redox cofactors, NAD+ being the oxidized form and NADH being the reduced form. Again, the only change in the structure occurs in the nicotinamide, with the rest of the molecule being a place for an enzyme to bind. Notice that for all three cofactors, the enzymes are recognizing an adenosine. When NAD+ is used, it catalyzes the oxidation of polar bonds, most often an alcohol or an aldehyde. NADH is produced in these reactions. Conversely, NADH catalyzes reductions of polar bonds and produces the more oxidized NAD+. Coenzyme A, often abbreviated COASH or COASH, is responsible for carrying acyl groups, like in fatty acids, and in this chapter specifically acetyl groups, a 2-carbon acyl group. Notice that again, adenosine is part of the structure. Acetyl-CoA is the entry point into the central metabolism. Again, our important molecules that we want to keep track of are all forms of ATP, which are used for most energy functions in the cell, oxidized and reduced forms of both FAD and NAD+, and acetyl-CoA because it can be metabolized in the citric acid cycle and the electron transport chain and converted to energy. ATP is the major energy currency of the cell, but phosphates carry energy in many forms. The phosphates on the left are actually higher in energy than ATP, so can readily phosphorylate ADP and make ATP. The phosphates on the right include ATP and ADP in similar molecules, and lower energy phosphates that can be made from ATP. Our goal is often to make the phosphates higher in energy so we can collect its energy as ATP. We'll see this in the citric acid cycle, but even more in glycolysis next chapter. To put our central metabolism in context, we will generally be talking about the path of energy all the way from a food that contains a major biomolecule to its conversion to acetyl-CoA. Once we get to acetyl-CoA, the catabolic pathways to energy are the same for all biomolecules. We go through the citric acid cycle, which is also called the Krebs cycle after the Nobel laureate who did much of the initial studies on the pathway. It's also known as the tricarboxylic acid cycle, or TCA cycle, because why not? I'll use citric acid cycle, and so does your book. The citric acid cycle is cyclic. It begins and ends with the same molecule, so it's actually what happens to the acetyl-CoA and the byproducts of the cycle that are important. Those byproducts take energy harvested in the citric acid cycle into the electron transport chain, so both pathways are connected. Again, the very big picture is that we will, for any biomolecules, break dietary forms into simpler forms that can enter our bloodstream, like fatty acids, glucose, and amino acids. Each of those pieces can be used in a variety of ways, but when they're used for energy, they become acetyl-CoA and are processed through the citric acid cycle and the electron transport chain. And those are the main focuses of this chapter. We're going to go into specifics about the citric acid cycle in a little bit, but the most important thing to know here is the overall reaction. For my purposes, you can ignore the PI, which is inorganic phosphate, the water, and the H+. Note that most of the reactants are paired. Acetyl-CoA comes in, 2 carbon dioxides and an unloaded CoA go out. 3 NAD+ go in, 3 NADH come out, FAD goes in, FADH2 goes out, GDP goes in, GTP comes out. In this cycle, the acetyl carbons are oxidized to carbon dioxide and all the redox cofactors are reduced. The carbons coming in bring the energy, and the energy leaves ready to be used as GTP or as NADH and FADH2 that can be processed for energy in the electron transport chain. The citric acid cycle is an example of metabolic regulation through allosteric enzymes as well. Citrate synthase, which is the first enzyme we'll talk about, is regulated by feedback with ATP. Since ATP is the end goal for the citric acid cycle and the electron transport chain, having lots of ATP slows down the cycle. The third enzyme, isocitrate dehydrogenase, has 2 allosteric regulators, one that's negative and one that's positive. The 2 regulators are NADH and ADP. Which do you think is a positive regulator, or that is, which of these do you think will make the citric acid cycle go faster? The citric acid cycle is a cyclic pathway, in case that wasn't obvious from the cycle part of the name. The citric acid cycle begins with acetyl from acetyl-CoA being joined to oxaloacetate, and the remainder of that cycle is oxidizing those carbons off and returning the structure to oxaloacetate. Many of the most important steps are oxidations. This is where we generate the cofactors NADH and FADH2 that can be used for energy elsewhere. The first step of the citric acid cycle is considered a condensation. We join two pieces together to get the acetyl-CoA carbons into the cycle. The second step of the citric acid cycle doesn't give us any energy, but it does posture us for getting energy at a later step. Moving the alcohol allows us to harvest more energy later, since we can oxidize that alcohol at its new location. It moved from being a tertiary alcohol to being a secondary alcohol. Step 3 is an oxidation and a decarboxylation. The oxidation is really important because it uses NAD+, meaning we need to have a supply of NAD+, and because it produces NADH, which can be used for energy if we have oxygen. Once the alcohol is oxidized to a ketone, reactivity that we don't talk about in this course allows for the carbon dioxide to easily be removed. This is important because it allows the neighboring carbon, which was tertiary, to become secondary and more easily oxidized. The fourth reaction is another oxidation. It's harder to see because it is actually a ketone that gets oxidized, which we rarely see. When the ketone is oxidized, it releases the carbon dioxide and is trapped as a thioester, which is a derivative of a carboxylic acid. Notice that the oxidation uses another NAD+ and produces another NADH. We're now at a total of 2 NADHs produced that will account for some of the energy we get from the citric acid cycle. The succinyl-CoA is trapped in a higher energy state from the oxidation in the previous step. Step 5 is the only step where an energy-carrying product, in this case GTP, is produced without oxidation. And that's only because the energy came from a leftover in the oxidation in the previous step. All the energy in the citric acid cycle comes from oxidation and is stored in reduced cofactors. After this step, we're at a total of 2 NADHs that have been produced in the cycle and 1 GTP. The next step is the first of a sequence of reactions that are really common in biology. An oxidation from an alkane to an alkene, that's then followed by a hydration of that alkene to an alcohol, and then followed by another oxidation. Again, this first step of that common sequence converts an alkane to an alkene. FAD is the preferred redox cofactor for oxidizing those nonpolar CC bonds. So going from alkane to alkene is almost always FAD. So we use FAD and we produce FADH2 in this reaction. Now we're at a total of 2 NADHs, 1 FADH2, and 1 GTP produced in this sequence. Hydrations are not oxidations. The alkene is just as oxidized as the alcohol, but it's hard to oxidize an alkene any further, but it's easy to oxidize an alcohol. So the hydration step at step 7 allows us to posture ourselves for an additional oxidation. Our final reaction is another oxidation, giving us a final NADH and restoring oxaloacetate, which we can now react with another acetyl-CoA. Again, don't memorize the individual steps of the citric acid cycle, but do know what the overall reaction is and what types of steps are giving us energy in the form of NADH and FADH2. Hint, it's oxidation. I just want to really briefly revisit this slide now that we've gone through the citric acid cycle. Again, our general goal when converting food into energy is to break it down and absorb the simple forms of each type of biomolecule into the bloodstream. Each type of simple biomolecule may be broken down into acetyl-CoA, which then gets processed in the citric acid cycle. This cycle is connected to our next process, the electron transport chain, by the redox cofactors NADH and FADH2 that are produced in the citric acid cycle. Again, 3 NADH and 1 FADH2 are produced in the citric acid cycle, and they will be used in the electron transport chain. Again, the electron transport chain is connected to the citric acid cycle because it processes the NADH and FADH2 from the citric acid cycle into NAD plus and FAD that can be used again, and it uses the energy harvested to produce ATP. The electron transport chain only works if there is oxygen to ultimately receive the electrons from NADH and FADH2, and the citric acid cycle only works if the electron transport chain returns NAD plus and FAD to it. The combination of the electron transport chain and the citric acid cycle, where carbons get oxidized to carbon dioxide and oxygen gets reduced to water, is essentially combustion, except broken down into little pieces so that we can get ATP from it instead of, fire. If you look closely at the diagram, you'll notice a few things. The first is that NADH and FADH2 enter the electron transport chain at different complexes, but both make the same CoQH2 cofactor. Also notice that the complexes 1, 3, and 4 pump H+ into the inner membrane space. You can see the H+ with the blue arrows coming through. That eventually gives us the energy to make ATP. These are the major highlights to look for in the next slides, in which we look at complexes that process electrons from each cofactor and how much H+ they reduce. The electron transport chain works because NADH and FADH2 electrons are relatively high in energy. These electrons could be used to reduce oxygen to water directly, but by making small transitions in energy at many complexes, we're able to harvest more useful energy as a gradient of protons, that is H+, and ultimately ATP. Notice again that NADH uses complex 1 to send electrons to CoQH2, while FADH2, which is lower in energy, uses complex 2 instead of complex 1 to send electrons to CoQH2. Again, the electrons are always moving from higher energy to lower energy until ultimately they combine with oxygen to make water. We need oxygen for the electron transport chain and therefore the citric acid cycle. The first complex, complex 1, oxidizes NADH to NAD+. That can be used in the citric acid cycle again. There are many redox reactions that happen within complex 1, beginning with electrons being transferred onto a flavin, like in FAD, and then to an iron-sulfur protein, and then to a coenzyme Q, abbreviated CoQ, which becomes CoQH2. In the process of this series of redox exchanges, complex 1 will pump 4 protons for every NADH. Remember that these 4 protons are only produced for NADH, since FADH2 does not send any electrons through complex 1. So again, highlights, complex 1 is responsible for oxidizing NADH, and it pumps 4 protons. Complex 2 only processes FADH2. It never does anything with NADH. Since FADH2 starts out as a flavin, it never sends its electrons to another flavin, like what happens to NADH electrons in complex 1. But it does send electrons through an iron-sulfur protein and eventually to CoQ to reduce it to CoQH2. Because this is a smaller drop in electron energy, complex 2 is not able to pump any protons into the inner membrane space. So now we've looked at complex 1, which takes NADH electrons and puts them onto CoQH2, and complex 2 that takes FADH2 electrons and puts them onto CoQH2. So we have the same redox cofactor for both complexes, they just reduce different pieces. NADH in complex 1, FADH2 in complex 2. Notice that complex 1 and 2 are never both active. It's always one or the other to make the CoQH2 that will bring electrons to complex 3. Because complex 1 pumps 4 protons and complex 2 pumps none, NADH will have higher ATP yields than FADH2. At complex 3, we have another cascade of reductions, bringing electrons from CoQH2 and restoring CoQ by moving those electrons eventually to 2 cytochrome C molecules. Cytochrome C is a heme-based protein that handles electrons by converting iron at the center of a heme from +3 charge to +2 charge. Since each reduction only handles a single electron, 2 cytochrome Cs are produced for every CoQH2. This complex is able to pump 4 protons for each CoQH2 that reacts. Complex 4 takes the 2 cytochrome Cs that come from complex 3 and then oxidizes them. With electrons it gains, it reduces oxygen to water and pumps 2 more protons into the intermembrane space. This is the end of the electron transport chain, but again, it only works if the electrons have someplace to go at the very end. We cannot use the electron transport chain without oxygen. For a summary of what happens in the electron transport chain, follow the arrows from either NADH or FADH2 through each complex in the top figure. NADH engages complexes 1, 3, and 4, while FADH2 engages complexes 2, 3, and 4. By looking at which complexes pump protons, we see that NADH is able to pump 4 protons at complex 1, 4 protons at complex 3, and 2 at complex 4, for a total of 10 protons. Similarly, FADH2 is only able to pump 4 protons at complex 3 and 2 protons at complex 4, for a total of 6 protons pumped per FADH2. So again, NADH2 is responsible for pumping 10 protons with its electrons, and FADH2 is responsible for pumping only 6. In the ATP synthase complex, all the hydrogens that are present in the intermembrane space can pass back through like water through a water mill. The energy of the hydrogens passing back is used to physically pound a phosphate onto ADP and produce ATP. This overall reaction gives a yield of 1 ATP for every 4 protons. Since we know how many protons per NADH or FADH2 and how many ATPs per proton, we can determine how many ATPs an NADH or FADH2 are worth. NADH pumps 10 protons, and since 1 ATP is produced per 4 protons, 10 divided by 4 gives a yield of 2.5 ATP per NADH. Similarly, FADH2 pumps 6 protons. 6 divided by 4 gives a yield of 1.5 ATP per FADH2. Notice on this slide that these conversions have been taken one step further. If NADH is worth 2.5 ATP, the citric acid cycle produces 3 times 2.5 ATP from the 3 NADHs for a total of 7.5 ATP. The citric acid cycle also produces 1.5 ATP from the single FADH2 that's produced, and a GTP is also produced in the citric acid cycle that has about the same level of energy as an ATP. The citric acid cycle and the electron transport chain together then produce a total of 10 ATP for every acetyl-CoA. So again, acetyl-CoA is worth 10 ATP, NADH is worth 2.5 ATP, and FADH2 is worth 1.5 ATP. ATP is produced from the catabolism of every major biomolecule in catabolic processes, but it is also the fuel we use to power all sorts of work, from powering anabolic pathways to allowing our muscles to contract. So again, we want to keep a good track of how much ATP some of our most important molecules are worth, because they give us a good way to track how much energy they are worth overall. In case you missed it before, acetyl-CoA is worth 10 ATP, NADH is worth 2.5 ATP, and FADH2 is worth 1.5 ATP. Most of the oxygen we breathe in is used in the electron transport chain, but the rest becomes reactive oxygen species. We use these reactive oxygen species in our white blood cells to destroy whatever they're attacking, or in our peroxisomes to metabolize molecules that don't have dedicated pathways for degradation, for example drugs, which we talked about with the grapefruit effect. We only want these in really dedicated places because reactive oxygen species can also do all sorts of damage to our own bodies, which is why it's important to avoid exposure to reactive oxygen species. Common avoidable sources are things like UV light, industrial pollutants, and smoking. Don't smoke.